So we know now that the brain controls appetite, right? And if people are obese, it's because brain pathways controlling appetite are not working properly. And what's really amazing about GLP-1 receptor-based therapies is that initially they were designed to treat diabetes, a disease of the pancreas, right? But we discovered in the past 10 years that these drugs also target the brain, and it is through their brain effect that they modify appetite and lead to weight loss. So we really need to understand how they work in the brain, how they access their brain targets to produce weight loss. So the understanding of weight loss biology in humans is really founded of what we understand in animal models. So I think it is very likely that our findings will translate into humans. And we know, for example, that drugs that target both the GLP-1 and the GIP receptor in humans are more efficient to produce weight loss, but we don't really understand how. So this is providing a mechanism to understand how this is working and then help us design the next generation of drugs to further enhance these effects. So we're really talking about the mechanism of action here. We know that drugs that target both receptors are more efficient in women or in humans, but it's understanding how. So we could use a number of human brain imaging strategies to determine if the same mechanism as the one we identified is also present in humans. For example, we could radio-labeled semaglutide and see if it enters the brain more efficiently in combination with a GIP receptor agonist. I think it's important to understand that GIP receptor agonism does not do the same as GLP-1, and it's really GLP-1 that produces the weight loss effect. And what we think is that GIP will change the population of cells in the brain that are exposed to GLP-1. So it's really a combined effect. They work together to produce weight loss. So what we find is that GIP receptor agonism enhances the entry into the brain of GLP-1 receptor weight loss drugs. So it means that different populations of neurons in the brain will be exposed to the drug. So it can mean maybe less side effects. It can be maybe more satiety. It can also maybe mean that brain areas involved in other diseases, we can think Alzheimer's disease, might be better exposed to the drug. So we need to better understand the implication of this for the side effects of GLP-1 receptor-based therapies, but also for other diseases.